Welcome to another ALS Expert Talk. We're bringing these to you because you're in your living room, and we know we're going to come to your living room. So those are the kind of things that we think about. We think about your needs, as deep as they might be. We're thinking about them. So with your inspiration, we find these inspirational speakers. They just come because they want to serve the community, and we have a way to let them do that. So I just want to say thank you to everyone who came tonight, because we've got something to learn. Over to you, James.
Well, thank you. Welcome back, everybody. For those of you who have been here before, welcome again. And for those of you who are new, welcome. I'm going to introduce Indu right now. I believe she has a few announcements she'd like to make. So Indu,
Yes.
Go right ahead.
Yes. Thank you, everybody. And as McFinn said, we are a family. And what we wanted to tell you is that we've actually started an awareness campaign for ALS. And if you go to our website, there is stories. You'll see a tab called Stories. And I want to thank many of the people living with ALS have participated in this campaign. So this campaign is really, we get to know where you are. Say you're in Colorado or Texas in some city. We contact the news in that area, in that local area. We have our PR and marketing team, and we cover the story. We cover who you are and what is ALS about. So I want to especially thank Paul Miller, Tommy Culpepper, Gwen Peterson, Brian Andre, Lisa Cross, Austin, Edie Friedman, some of the few people who have actually participated in this. And we've seen this as being a very, very impactful way of getting our stories out. So if any of you here are interested, please let us know. You can send us an email at info@everythingals.org. And if you would like to be covered in your local area in the news, either in TV or in articles, please let us know. And one thing is that we are covering people who are in our study. So you have to be in our study, in our speech study, and we would love to see more faces there. So with that, I'll pass it back to James.
Wonderfully said. And it's a really cool opportunity. And I just wanted to take this moment to allow people to know that on any part of the everythingals.org page, no matter which heading you pick in the top, if you scroll all the way down, again, doesn't matter which page you're on, and you click the little follow us button where it has the little YouTube icon, it's going to pull up for you our page, and it's probably going to give me an automatic playlist right there. Perfect. You can click that subscribe button, and all of the videos we have that we do every 2 weeks will be right here for you in case you miss one. So that's just there for you as well. With that being said, though, I do want to talk about our guest speaker for today, Dr. Charles Brenner, and I'm going to read a little intro about him. Dr. Charles Brenner is the Alfred E. Mann Family Foundation Chair in Diabetes and Cancer Metabolism in the Department of Diabetes and Cancer Metabolism. His laboratory focuses on disturbances in nicotinamide adenine dinucleotide, NAD, the central catalyst of metabolism in diseases and conditions of metabolic stress. Among his most significant discoveries, he identified nicotinamide riboside, NR, as an unanticipated vitamin precursor of NAD. He also developed a quantitative I've always butchered this word, metabolomic technology that allowed him and others to discover that the NAD system is disturbed by many diseases and conditions, including diabetes, cancer, and amyotrophic lateral sclerosis. In this talk, Brenner will review what NAD is and does, what is known about disturbances of NAD systems in ALS, and what data from mice in early stage clinical trials may mean for people living with ALS. With that being said, I would like to introduce and welcome and turn things over to Dr. Charles Brenner.
Thanks very much. I'd like to thank my hosts and thank your outstanding organization and community for doing what you do and for asking me to participate in in this small way. So I'll see if I can share my screen, and I'll probably ask you in a minute whether you can see my pointer. Let's see, first I have to share the screen. And here's the talk. And here is slideshow. And can you also see my pointer?
Perfect.
Yes.
Beautiful. Okay, thanks. Now, if you're kind of an experienced Zoomer, you can probably put it on speaker mode. And rather than looking at the whole gallery, you can just look at one person that is the speaker, and you can park me in the lower right-hand corner of your screen if you know how to do that. That
There
would be
we go.
great. So, I'm Charlie Brenner. Today is my 2-year anniversary at City of Hope in LA County. I'm originally a Bostonian and moved around throughout my career. I'm a biochemist, and I'm one of the people in the world that knows the most about NAD, the central catalyst of metabolism, and that's mostly what I'll talk to you about today, and how I think it's actually a real key to understanding the pathology of ALS and potentially therapeutic approaches, of which I will name 2. And so let's see if I can advance the slide. I do have disclosures. I developed the intellectual property around nicotinamide riboside, which I will be mentioning in that context. I'm the chief scientific advisor of Chromadex, which developed it, Juvenis, which is a new company that is trying to use NR in skin, and I'm a co-founder of a company in the cancer space. This is a view looking north out of our laboratory window at City of Hope. You can see Mount Wilson in the background, and the City of Hope Library in the foreground. And so I'm going to start that this question is going to make the most sense for the few of us that are watching this talk from here in Los Angeles. Does anybody remember what the weather was doing exactly 8 weeks ago today? It was June 22nd, so it was the first full day of summer. And it was a little bit of a surprise for us, because we woke up that morning to rain. So this is a view looking north along what's called the San Gabriel River in a town called Pico Rivera. And there was a woman that was peacefully walking her 2 dogs that morning, and she was struck by lightning, and she and the 2 dogs were instantly killed. And this leads me to ask a question of all of you that will get you thinking about what metabolism is, which is, what's the difference between a living thing and their inanimate body? And the answer is metabolism. That is the life force. And just as those of you who own an electric vehicle, or know somebody that owns an electric vehicle, know that there is a huge battery pack in a vehicle that has wiring, some type of metal wiring, copper wiring typically, that goes between the battery and the transmission system and the cooling system and all the other steering system and all the other systems in a car. And the car cannot move without the flow of electrons. It turns out that no form of life can move without the organized flow of electrons. And the wiring in living things is not copper wire. The wiring are coenzymes. And the 4 most important coenzymes, those that we, in fact, focus our laboratory on and are focus my entire professional life on, are called NAD coenzymes. NAD stands for nicotinamide adenine dinucleotide. So you'll sometimes see NAD plus, other times you'll see NADPH, NADH, NADP. These are the 4 NAD coenzymes, and they not only allow us to convert everything that we eat into ATP, our biological energy, but they run and all of the organs in our bodies. I like to say metabolism converts everything that we eat into everything that we are and everything that we do. In fact, metabolism also allows us to repair and maintain our whole body. So all of our vital functions in every organ depend on NAD coenzymes, including replicating and repairing DNA, controlling free radical formation and action. That's primarily a function of NADPH. And so metabolism is the life force. And now the second thing, I'm credited with discovery of the nicotinamide riboside kinase pathway to NAD. So one of the ways that cells make NAD. But the other thing that I think is just as important is, as was said in the introduction, we developed the methods that allow us to determine whether the NAD system is stable or the NAD system is being disturbed. Sometimes I will say the NAD system is under attack. And in different tissues, different types of metabolic stress or disease processes can cause the NAD system to come under attack. So in human beings, we've shown that alcoholism attacks the human the human liver NAD system. We've shown in mice, and I believe it to be true in people as well, that overnutrition or overweight and type 2 diabetes disturb the NAD system in the liver. Sufficient noise to produce hearing loss disturbs the NAD system in our cochlea, in our hearing organs. We know that sun and oxygen damage, like DNA damage caused by the sun and reactive oxygen species damage, so these are inevitable forms of metabolic stress, they attack the NAD system. We've shown that postpartum is the mother of all metabolic stresses, that models of heart failure disturb NAD. I'll have quite a bit to say about neurodegeneration. We showed a couple of years ago in the beginning of the coronavirus pandemic that infection and inflammation disturb the NAD system. We have a project I won't tell you about today about certain types of cancer, and then it's widely reported that in the course of aging, the NAD system comes under attack. Although I sometimes wonder whether aging is merely kind of integrating all of these types of metabolic stresses such that there are discrete events, or there may in fact be time-dependent events as well that disturb the NAD system. So one of the technologies that we developed is this vitamin called NR, which is over here in the, I guess that's cyan, this kind of bluish green aqua color. And NR can restore and boost NAD such that these metabolic stresses can attack the NAD system. NR can replete or fill up our NAD stores, NADPH as well, and that can sort of allow us to optimize cellular health and resiliency. And you'll see some examples of that here in this talk. So just to name those 4 NAD coenzymes, they're NAD+, NADH, NADP+, and NADPH. And just for those of you that haven't spent years studying biochemistry, the basics of fuel oxidation, converting stuff that has calories, protein, fat, and carbohydrate into ATP, that's kind of the NAD to NADH process. Actually, it's NAD is used to oxidize these fuels generating NADH, and then NADH at the intermitochondrial membrane distributes the electrons in order to make ATP. And NADH is also used to maintain our blood glucose and to generate ketone bodies. Then NADP and NADPH are largely used in anabolic processes and making things like DNA and RNA precursors, making steroid hormones like estrogen and androgens, generating fatty acids, and also, very importantly, for ALS, doing detoxification of free radicals. All of this involves electron capture by the plus forms and generating coenzymes that are called the H, the reduced forms, and that NADH or NADPH is carrying a high energy electron that can do biological work like help us make ATP or detoxify free radicals. So I won't spend a lot of time, talking about organ systems other than the heart other than the brain for obvious reasons. But let me just show you something kind of interesting from the heart, because it's a good lesson for us, and it actually tells us some of the differences between NR, nicotinamide riboside, and other NAD precursors. So before our lab came along, the received wisdom was that animals can make NAD inefficiently from an amino acid called tryptophan, or from two forms of vitamin B3 called nicotinamide and nicotinic acid. Nicotinic acid is sometimes called niacin. And we discovered that there's this other gene pathway that's encoded by NMRK1 and NMRK2. This is the NMRK2 gene here, and this shows the gene activity, the gene expression level going up about 40-fold in a disease process in the mouse. So look at what's happening in this disease process. This is a mouse that is going to develop heart failure. It has a 40-fold induction of the nicotinamide riboside kinase 2 gene, while it has a 40% drop in expression of the NAMPT gene. So the NAMPT gene is the gene that is used to convert nicotinamide into NAD coenzymes, and the NR genes, NMRK1 and NMRK2, are used to convert NR. So here in a disease process, the failing cardiomyocyte, the failing heart cell, is losing its ability to generate NAD with nicotinamide and is gaining the ability to generate NAD from the NR pathway. And it turns out that's important. And if you give, and I won't go through all of the data in gory detail, but in the right hand of these 4 bars, like this is a left ventricular ejection fraction, and this is fraction shortening. So these are measures of heart function. These are these mutant mice that have a low ejection fraction and a very terrible fraction shortening. If you give them NR in their oral diet, you largely correct the heart problem. The fraction shortening gets much better, the ejection fraction gets better, and all of these parameters get better. And you can't rescue the NAD problem with nicotinamide. Why? Because the nicotinamide gene pathway is going down in the failing heart, and the nicotinamide riboside kinase pathway is going up. It actually costs less ATP to make NAD with NR than it does with any other NAD precursor. So this is some of the value in the NR technology that we initially discovered at Dartmouth and have been working on ever since. So NR is kind of a special NAD precursor. And you can see a failing heart that is losing NAD and is having a bioenergetic crisis. And those of you that follow ALS know that there's a bioenergetic crisis and a redox biology crisis in ALS. They're having a crisis and they're losing expression of one NAD synthesizing pathway, which may in fact be part of the pathology, but they're gaining expression to make NAD from NR. Now, you said, okay, that's fine, but I've got a couple of problems with that. And you were all telling me, first, that's the heart, Brenner, and second, that's a mouse. So, okay, so I'm going to move towards to the brain now. And you can see that I'm going to go from something that seems very unrelated to, I'm going to take you all the way to human clinical trials in the course of the next few minutes. So this is a central brain injury model where excitatory stimulus, NMDA, is basically leading to a brain lesion. A lot of times, as you know, researchers are very impatient. And so they want to generate a lesion, not even necessarily with a genetic model, but with maybe a toxin or a brain poison. And then they want to see, what they can try to cure that or prevent that with. And so this is an NMDA toxicity model in which the NAD level, if you can follow my pointer, the NAD is being cut in almost half. It's like losing maybe 40, 45% of the NAD level in the brain. And then look at what is happening to the expression of the NMRK1 and the NMRK2 genes. Once again, just like in heart failure, where NMRK2 was the most highly induced gene, NMRK2 goes from being almost not expressed at all to being significantly expressed at 24 hours. And the NMRK1 gene is doubled in 3 hours and, increased by something like, this is a funny scale, this is like half to 2.5. So it's increased by about 5-fold in 24 hours. So once again, the NMRK1 and 2 genes, the NR pathway, is getting upregulated when NAD is under attack. And sure enough, NR protects these brains from the lesions that are induced by NMDA. So that was nice. So that was a kind of a protective model in the heart that goes with NR kinase increased expression. Now we have a protective model in a mouse brain. And now I'm going to make it much more relevant to ALS because this is a knock-in or a replacement of human superoxide dismutase Gly93 alanine, which is, as you know, is a disease allele of superoxide dismutase, which is there to control reactive oxygen species. And this leads to morbidity and mortality in the mouse. One of the ways that you can measure that is you can look at the age at which they've already lost 10% of their body weight. We don't even, we don't like to see animals in discomfort and pain. And you can start seeing the ALS progress in a mouse with body weight loss. So you can see the age at which they've lost, a growing mouse is increasing its body weight, right? But a sick mouse is going to get down to 90% of its high watermark of body weight. It's going to get to 85%, et cetera, et cetera. And you can see that the control mice, actually, they're SOD knock-ins. So these have a human, disease gene in them, are down to 90% of their body weight by 145 days or so. And those that are treated with nicotinamide riboside are protected. So they're not as healthy as a wild-type mouse, but with a very impressive p-value. And you can see the multiplicity in the number of animals that are tested. This whole population is doing better than this population. So NR is pretty protective. And that's not the only data that are in this paper, Harlan et al. But this paper, Harlan, shows that de novo pathway by which animals can make NAD from tryptophan is actually messed up in ALS. So even though we're knocking in superoxide dismutase in the disease model in the mouse, they're losing their ability to make NAD from an endogenous pathway such that they greatly benefit from this vitamin activity of nicotinamide riboside that we identified. And I gave you my the financial disclosure about that. Of course, this has been commercialized as a safe over-the-counter molecule. So now of greater relevance now let's go into human beings. And if we start looking at the redox potential, which is a measure of the NAD to NADH ratio in human brains that is measured by magnetic resonance spectroscopy, you can see that there is a decline as a function of aging, suggesting that in the course of human aging, there is an NAD-related bioenergetic problem that potentially could be addressed by supplementation or some type of a drug approach that would affect either the total levels of NAD or the redox state of NAD, which is, as I said, it's the NAD plus to NADH ratio. So this is becoming, I would argue, more and more interesting. Now, this is not my figure. This is a review from Fu et al. In Nature Neuroscience that shows a number of different types of neurons in Parkinson's and Huntington's, in Alzheimer's disease, mostly affecting pyramidal neurons, and of course, in the debilitating disease ALS, it's mostly motor neurons, spinal cord neurons, and neurons in the brain stem that are being most profoundly vulnerable in these degenerative conditions, where there's clearly a problem in cellular bioenergetics, resiliency, control of reactive oxygen species. And so it was in light of all of these data from all of the model systems, from observations of human aging and human ALS disease genes knocked into mice, that De La Rubia et al. Completed the first small study. 20 patients finished this randomized control trial, placebo-controlled trial, involving nicotinamide riboside. And they scored an improved ALS functional rating scale. I kind of feel like all three of their of the things that they report in this paper in ALS and frontotemporal dementia, volume 20, I feel like all of these are related functional metrics of ALS, and they all basically declined in the placebo group. So in 4 months, these patients were of a severity in which one was able to measure a decrement in their performance over the 4 months, and those that were on the active had improved functioning. So they did their statistics primarily comparing the treated group versus the placebo group, because so they're incorporating the benefit from the treatment as a delta against the decrement in the placebo group. And I will point out that if you read this trial, it's nicotinamide riboside plus pterostilbene. Pterostilbene is a natural product that is found in blueberries. We can talk about it. I don't believe there's any credible reason why pterostilbene had a functional role in the activity of the treatment. In fact, the human data on pterostilbene indicates some, toxicity to metabolic conditions in that it raises low-density lipoprotein cholesterol, which is a known, risk factor and in fact driver of cardiovascular events. But for reasons that related to business, largely, the ingredient of nicotinamide riboside was combined with pterostilbene, and but it got this positive result in the small trial. And so there's a follow-up in Norway with 380 patients that has been registered on clinicaltrials.gov. They're going to be looking at a 1-year endpoint. They are also including pterostilbene in the treatment group. They're doing 2 different amounts of NR and pterostilbene. One of them corresponds to 1 gram of NR per day, and the other at, I believe, 1.5 grams a day. But when you, in this particular product, that's a combination NR plus pterostilbene, you end up getting a lot of pterostilbene, which is probably going to lead to cholesterol problems for the patient population. I've actually spoken to the people who are doing that study, and they felt that because the small trial by De La Rubia was NR plus pterostilbene, they didn't have a way to not include the pterostilbene in their larger trial. Now I'm going to shift gears a little bit, and I'll tell you about some data in which the trial design were published by Vusic et al. In BMJ Open, and the data have not been peer-reviewed, but the data are being made available on a company website. And I have no financial relationship with this company. It's called Clean Nanotechnology. And what they're using is a gold nanoparticle called CNM-Au8. Au is gold. It's a gold nanoparticle that captures electrons kind of in the same way that NAD+ does. So if one of the problems in ALS is that the NAD/NADH system is not effectively capturing electrons, and these free electrons are part of a problem of Ross generation, then a molecule like CNM-Au8, by elevating the NAD plus NADH ratio, could be potentially helpful. There is evidence, in fact, that this is a way that you can raise ATP in cells that are challenged to generate ATP. Now, this was a somewhat underpowered study. It was 36 weeks. And you can see that they're using a performance metric, a motor performance metric called Munix. And the limb onset subset of Munix in particular shows that the active or the treatment group is generally doing quite a bit better than the placebo group, but the 95% confidence intervals cross the 0, which means that from this particular parameter, they don't have quite the statistical power to put a flag on the ground and say, this is absolutely a positive result in terms of meeting their primary endpoint. But I want you to look at some of the additional endpoints that were pre-specified, but as exploratory endpoints, such as the disease progression. So if you look at percent event-free in the clean CNM-AU8 trial, you can see very few of their patients are event-free in the placebo group, but the folks that are on CNM-AU8 are largely event-free. And so this achieved a very impressive p-value of p.0125. And even maybe more remarkably, and again, these data you can see yourself on the CLEAN website, is that their trial was conducted in this 36-week period in which the patients were subject to a double-blind, placebo-controlled trial. But there's a follow-up period that went, 36, 70, 80, 90 weeks beyond that. You can see some data up to 120 weeks total. And you can see that there's a long-term survival advantage where this is actually, these are living people. And this is the Kaplan-Meier curve of folks that are not, the folks that are making it are in these lines, but only 30% of the folks that were enrolled are still alive, if I am reading the study correctly. So this is a pretty remarkable preliminary result showing that a 36-week exposure to CNM-AU8 not only nearly showed in a small trial, showed a performance benefit, but it seems to clearly help people longer term. So they're following up with something called the Healy ALS trial, which according to company statements should show top-line results this quarter. So we can all look forward to seeing how that comes out. So in conclusion, and I'll be happy to take questions, the NR pathway, so NAD first, NAD coenzymes are, the central catalyst of metabolism. Metabolism is, basically it's the life force. And the NR pathway is upregulated when NAD comes under attack. And that the ALS pathology appears to involve impaired NAD-dependent bioenergetics and ROS control, and that both NR and CNM-AU8 are promising approaches to treatment. CNM-8 can only be accessed through clinical trials. Nicotinamide riboside has been tested for safety in the general public, in the aging public, in the overweight public, in the context of NR and pterostilbene, in ALS patients. NR is safe up to 1 to 2 grams per day. And so this is something that we know anecdotally members of the ALS community are trying. I'm always asked about a dose. I'm a PhD scientist. I'm not your doctor, but I will state that most clinical trials are done in the 1 to 2 gram range. You can see that the Norway ALS trial is being tested at an effective dose of 1 to 1.5 grams of nicotinamide riboside per day. The safe stuff that has the longest safety dossier comes from Chromadex, is called Niagen. It's in a blue bottle. And I'd be happy to take questions from everybody on the call. Thank you so much. We can't hear you.
You're on mute.
There we go. Sorry about that. Saying thank you for that presentation. Myself and Pooja, who's a student ambassador, we will be fielding the questions that were asked in the chat for you. I'm going to start off with a pretty simple one of, can you perhaps explain the difference between NAD and NAD+?
NAD basically encompasses the 4 NAD coenzymes. Frequently, NAD is used as NAD+, but in a larger context, it includes 4 related NAD coenzymes.
Got it. And then our next question is, are nicotinamide levels similar in those with both sporadic and familial ALS?
Not known. And we don't have access to, human brains, right? So, so I don't even know whether anybody has done a study to look at blood NAD in ALS patients. That's something that would be of interest. I was personally somewhat surprised when we did our first study of individuals with a mitochondrial myopathy, because they've got a mitochondrial disease presentation that is largely in their muscle. And so I wasn't confident that we would see a defect in their blood NAD, but there are people with mitochondrial myopathy that are walking around with low blood NAD. So low blood NAD would be informative, but not seeing low blood NAD doesn't mean that there isn't a defect in the ALS motor neurons. I think there probably is a defect.
More research will be needed. Generally speaking, is it preferable to take NR, NMN, or NAD+, and does the answer change for neurological diseases like ALS?
Easy question. NMN and NAD don't really make sense as NAD precursors, because you ever heard a chemistry lesson here, but they have phosphates on them. So NR is the largest piece of NAD that can get into cells, and then cells use the NR in order to generate NAD. NMN, and I know there's a very vocal person out there, kind of influencer out there, that is always talking about NMN. NMN is NR with a phosphate group on it. We and many other groups have shown that the NMN gets converted to NR before it can go into cells. NMN is not further down the pathway than NAD. Similarly, if you were to eat raw food that has lots of NAD and NADH and NADPH in it, your digestive process is going to break the NAD down to NR, nicotinamide and nicotinic acid, in order to build up your NAD. So NMN doesn't make sense as a supplement. NAD, NADH, NADPH don't make sense as a supplement. They just don't. I can anticipate some of the other questions, and I can read some of them here. Liposomal NR, there's no safety dossier. There's no safety dossier on it. I don't really know who's making it, what's in it. You don't know who's making it or what's in it. There's no safety dossier. Intravenous NAD, again, doesn't really make sense because it's going into the bloodstream. It still has to break down into NR in order to get into cells. There's no safety. There's no regulatory structure that would know what you're getting if you try an IV NAD infusion. It's not safe. It's not recommended. So
James, do you want to go ahead?
Yeah, so that next question actually was exactly that. So you saw that one coming. What is the PK of NR, and how much escapes the microbiome as NR in circulation?
in humans, we don't know really the answer to that question. We know that there are some misleading and somewhat depressing interpretations of mouse experiments. Like there are mouse experiments that have been interpreted, and you go to the internet, I mean, this, I don't have to tell this community about, Dr. Google, and the ridiculous things that you can learn from Dr. Google and Dr. Facebook and Dr. Instagram. But even in PubMed, there are papers that claim that all of the NR is disassembled in the liver or in the gut, and then the gut or the liver is just circulating nicotinamide or nicotinic acid. There's no value of NR. That doesn't actually make any sense, because I showed you data in which the mouse has a failing heart. It's losing expression of nicotinamide, the nicotinamide pathway. It's gaining expression of the NR pathway, and oral nicotinamide doesn't do anything for that mouse, but oral NR does. So that means oral NR is available to the heart. So that means that oral NR is not being disassembled in the gut or the intestine. There are positive signals now in an ALS trial, in a Parkinson's trial. I'm aware of a positive cerebral brain flow signal for NR in a mild cognitive impairment trial. So clearly, some of the oral NR is available to the brain. And I can't quote you the percent that's available, but you don't have to worry about that. If the trials are done at 1 or 1.5 grams of NR a day, that's probably where you want to be.
Great. Our next question is, are you familiar with Hyundai Oslinder's work using NMN to boost NAD and its impact on upper motor neurons?
Not really, but NMN is basically going to act as NR, as I said. So if you provide NAD itself or NMN or NR, in fact, the data were in my talk. In the NMDA model, where you generate a lesion with NMDA, we tested NAD, and it's in a paper called by Vaur, V-A-U-R et al. In 2017. It's on my website. And NAD acts as NR, is blocked by the same thing that NR is blocked by. NMN is also blocked by the same thing that NR is blocked by. So NMN has to be converted into NR before it has any activity. So it's kind of annoying that there are people out there that don't know this. The cancer field figured this out by around 1980. In fact, the virology community figured this out by about 1980, 1985. So there are antivirals like AZT that are nucleosides. They have to act as nucleotides, but you can't deliver the compounds with phosphates. Literally any competent biochemist could tell you this.
That's good to know. Do you believe Elysium basis? Do you believe Elysium basis is a good product for NAD?
I don't have a lot of comments on that, since Elysium basis, was at one time, got their nicotinamide riboside from Chromadex, which developed the safety dossier, and they sort of used the Chromadex safety dossier to try to get regulatory approval for Elysium basis. They add the terastillabine to it, which I don't think has a credible mechanism of action. So it's not a product that I would recommend.
Going off of that, would you recommend terastillabine with
No.
NR?
There's no credible basis to make upon for terastillabine as being important in in ALS. That company that you mentioned claims that it's a SIRT1 activator, but there's a whole lore about sirtuins as longevity genes. This is basically, it's a longevity gene in yeast. It's not a longevity gene in animals. And pterostilbene doesn't even activate SIRT1 anyway. So no, I don't think that there's any reason for pterostilbene other than De La Rubia et al., the Spanish group, initially got their NR from Elysium, so it was already an admixture of pterostilbene and NR. But if you look at what pterostilbene does in people, and you follow blood lipids, it's doing some things to blood lipids that that people, you know, don't really need.
If PALs have a problem with NAD capturing electrons, how does this affect ATP output, and what does that output mean for those with ALS?
You need the NADH system to generate ATP, right? And so neuronal function is a highly bioenergetically demanding process. And so this gold nanoparticle, you know, it's interesting that it, you know, to some degree, it sounds like it's almost competing with NAD, but it's the mechanism of action is that there's an electron capture problem, a redox problem in the ALS motor neuron, and it's presumably on that basis that it's having efficacy. You basically, you know, it could have multiple targets, multiple mechanisms of action, but from the data that I've seen, I interpret the function of CNMAU8 as a electron capture, you know, molecule, and it appears to be quite safe in humans. So I'm pretty encouraged by the small trial, and I'm looking forward to the Healy ALS trial that hopefully they'll be announcing some results on soon. And I have no financial interest in that whatsoever.
All right, we're looking forward to hearing that as well. Our next question is, can you talk a little bit about the role of nicotinamide in microglia activation?
Interesting. So NR is an anti-inflammatory compound as well. So the neuroinflammation is generally a part of most of these neurodegenerative disease processes. And so there's a potential you know, set of targets in the, you know, activated microglia and astrocytes in which the boosting NAD could be basically be kind of calming a kind of cytokine or inflammatory storm. In fact, the antiviral mechanisms of action also appear to involve anti-inflammatory functions that depend upon NAD. So that's a really good question.
Sorry, we got a lot of questions right there. What is the most effective means to correct NAD issues in your opinion?
NR, oral NR. I don't think that there's anything else that's been shown to be safe. I mean, there classically there's niacin, right? So, but the problem with niacin is that nicotinic acid goes through a gene called NAPERT, and NAPERT is not highly expressed in neurons. And then the NAM pathway declines in many diseasing conditions of metabolic stress. So we think that this is why nicotinamide riboside is the kind of preferred and potentially most effective NAD precursor, especially in diseases. So I would argue that NR is potentially the best way to boost and restore NAD in a disease like ALS.
Going off of that as well.
Obviously, ask your doctor, but I guess these slides and presentations will be available to people. And there's actually a practitioner website as well for True Niagen. I think it's md.trueniagen.com, and medical practitioners can get a higher dose for their patients. They can get 500 milligram capsules for patients through md.trueniagen.com.
In addition to having people speak with their doctors, are there any products that you recommend for purchasing NR?
I told you the only safe, I mean, the nicotinamide riboside that has by far the biggest and most extensive safety dossier is made by Chromadex. It's called True Niagen. It's in a blue bottle.
Perfect. And we want to be very respectful for your time, so that this will be the last question.
Okay.
CNAU8 and NR sound like very promising therapies. And we believe that you said CNAU8 trials results will be available later this quarter. How soon do you expect NR might be available for ALS patients if the Norway trial is successful?
You always want to go to clinicaltrials.gov and see what the folks running the trial are saying about recruiting. And, a lot of times it's radio silent, right? But it may say that they're still recruiting, or it may say that they've completed their recruitment. You can't get a result faster than you can get it, right? Because, if it's a blinded study, everybody may still be blind, right? And you may not know which patients got which pills. So I have to say, I don't know when the Norwegian ALS trial will complete and be available, but generally speaking, clinicaltrials.gov generally has the latest information that are publicly available.
Well, with that, I would like to thank you for a very informative presentation. The next part of our little forum is the open kind of ended. Anyone can speak. You're more than welcome to stay. We understand you're very busy, so no pressure for you.
I'm happy to stay for a little while. I like to, answer questions on Twitter. Again, I'm not your doctor, but I'm pretty good at reading papers and kind of telling BS from things that are solid. And I don't like it when people exaggerate, cure disease in mice that can't be translated. And I'm very passionate about ALS and finding neuroprotective treatment approaches that will work. So it's really been an honor to meet this organization. You can learn more about my lab at brennerlab.net. You can ask me questions on Twitter at Charles M. Brenner. And this was fun. Thank you so much.
Well, thank you very much.
Charles. Charles, I'd like to ask you a question. Why is ALS so important in your life?
It's the people that I've met. It's the trying to understand what superoxide dismutase is doing. I've been thinking about that ever since I was a postdoc. So I don't actually have ALS publications, but I'm happy to have contributed to the NR technology, which seems to be potentially making a difference for some people. So I'm very, very happy to meet you folks and to support what you're doing.
Well, we really appreciate that you would choose ALS and be so dedicated to it. What did you think of our group questions this evening?
Great, really appreciated them.
Good.
By the way, blueberries, somebody said, how could blueberries raise cholesterol? They don't have cholesterol. Blueberries don't raise cholesterol. have as many blueberries as you want. What happens, and then this is something I talk about on Twitter quite a lot. There's a lot of natural products that are in foods that are great, right? Curcumin, it's wonderful, and there's phytochemicals that, and lycopenes and so forth that are part of a balanced diet, the colorful fruits and vegetables, whole 9 yards. Then
Charles?
a lot of
Yep. Go ahead.
a lot of times people have create a colorful theory about what their favorite compound does. Like people attributed what's called the French paradox, and they have better cardiovascular health than Americans, right? And so somebody said, oh, maybe it's because they drink wine. And then maybe it's the resveratrol in the wine. And generally, those stories turn out to be fantastical. And fantastical is not a good thing in science. And the resveratrol story is fantastical. Pterostilbene is part of the resveratrol story. If pterostilbene were a SIRT1 activator, then it would have lowered cholesterol in people. But pterostilbene increases low-density lipoprotein cholesterol in people, not healthy. And so the whole thing around resveratrol and pterostilbene is really a very persistent fiction, and it should not be, it should not be infecting your community. And it does not look like a healthful compound to take 50 milligrams of a day.
We needed to hear from you today, Charles. Thank you so very much.
Thanks.
And now,
All right.
yeah, please stick around and you can hear what everybody else is thinking on their